Welcome to the deep dive.
Mm,
Today, we're pulling back the curtain on something really quite fascinating.
we are.
The hidden science behind defining and, well, protecting precious plant varieties. Specifically, a spice that's, famously valuable,
hmm
saffron.
That's exactly right. We're going deep into the official guidelines used over in India. They're from the Protection of Plant Varieties and Farmers' Rights Authority.
Right.
And this document, it basically lays out the whole rigorous process that saffron varieties have to go through to get officially recognized
Yeah.
and protected.
So our mission today is to unpack this document, understand these, incredibly detailed steps,
Yeah.
and figure out maybe what surprising things it tells us about what makes one saffron variety truly different from another. Why does all this detail matter? What are they even measuring?
Well, because understanding these guidelines, it gives you this unique window into the sheer scientific precision needed to authenticate these plant varieties. It's really about ensuring quality, traceability too, and recognizing the innovation, behind both new and existing types. We're finding out what goes into proving a specific saffron is that specific saffron.
Okay, let's unpack it then.
Yeah.
The first sort of fundamental question is why? Why do they even have to test saffron varieties
Yeah.
so intensely in the first place?
It really all comes down to a legal framework. In India, it's the PPVFR Act from 2001.
Ah, yes.
This act gives protection to plant varieties, kind of like how patents protect inventions. So to register a new variety or even just document an existing one, it has to meet very specific criteria.
Okay.
And those criteria are proven through this standard test called DUS.
DUS, what does that stand for?
It stands for Distinctiveness, Uniformity, and Stability.
Right.
Distinctiveness just means the variety has to be clearly different, distinguishable from any other known variety by at least one important characteristic.
Okay, one difference is enough.
If it's a significant, stable one, yes. Then uniformity means the plants within that single variety need to be consistent enough in their key traits.
Makes sense.
And stability means those defining characteristics, they have to stay unchanged over repeated cycles of propagation, growing them again and again.
Ah, so it's really about establishing this reliable, unique identity, one that doesn't just disappear over time or like across generations.
Exactly.
And this applies to all varieties and cultivars of the saffron crocus, the Crocus sativus L.
Correct. All of them. Without this kind of verifiable identity, it would be, well, pretty much impossible to protect the rights of breeders or farmers' traditional varieties, or even just make sure consumers get what they think they're buying.
Got it. Okay, so let's say you've developed a new saffron variety. What do you actually need to provide to get it tested? What are the physical requirements?
Yeah, the guidelines are quite specific about the starting material. It has to be supplied as corms,
Corms.
which are basically the underground storage bits, like bulbs, where the plant keeps its energy.
Right, the corms. Is there a particular type or size they look for?
Yes, they specify corms weighing between 8 and 10 grams.
8 to 10 grams. Why so specific?
Well, that size range is pretty crucial. It helps ensure the corm has enough stored energy to support good initial growth. And importantly, lets the variety's traits express themselves reliably right from the first testing season.
That makes a lot of sense. You need a good, consistent starting point.
Precisely. And the corms themselves, they also have to be healthy, dormant, and completely free from any pests or diseases.
Okay.
And a really key requirement is transparency. They shouldn't be treated with any chemicals or biophysical agents unless that treatment is fully disclosed to the testing authority.
Right. Transparency is vital for a fair test. No hidden boosts.
Absolutely. And the material itself, it must show genetic purity and uniformity. As for how much? For a brand new variety, they ask for a minimum of 300 corms.
300. Okay.
But for varieties already considered, like common knowledge or traditional farmers' varieties, 150 corms is usually enough.
And what if a new variety passes the first year, but needs that second season of testing?
Good question. They use the first generation daughter corms, the ones produced by the plants from the previous year's test plots. It's a very direct way to assess the stability, how well the variety's characteristics hold up as it propagates.
Wow, that level of precision starts right at the source material. Okay, so where do these tests actually happen? And how long does this whole process take?
So for new varieties, the DUS test typically runs for at least 2 independent growing seasons.
2 seasons.
Yeah, and at 2 different locations.
2 locations as well. Why?
This multi-season, multi-location approach, it's critical. It helps verify that the variety's traits are actually stable and expressed consistently, even with slight environmental variations or, different weather year to year.
Right, so it's not just a one-off check.
Not at all. For the extent where farmers' varieties, though, one testing season is usually sufficient.
Okay, so they're not just testing it once in one place. They're really making sure it's stable and distinct over time and in different conditions.
Exactly. And the field test conditions themselves, they have to actively encourage the plant's normal growth. Let all their potential characteristics really show.
How do they set that up?
They have a very standardized plot setup. Each test involves a minimum of 100 plants per location.
Okay.
They're arranged in a specific way. 1 meter by 1 meter bed size, 5 rows, 1 meter row length, 20 centimeter row spacing, 10 centimeter plant spacing.
Wow, that's precise.
And this whole setup is replicated 3 times at each location.
3 identical plots at each of the 2 locations. That sounds really thorough.
It is. It helps account for any variability just within the field itself.
Right.
And there are restrictions too, like no using plant-promoting growth hormone.
Why not?
Because they want to observe the variety's natural inherent growth potential, not something boosted artificially. They also follow standard good agricultural practices and integrated pest management, or IPM.
Okay, controlling the variables so you're seeing the variety itself, not like the effects of external treatments.
Exactly that.
So once the plants are growing, what are they actually looking for? How do they measure and record everything? It must be a huge amount of data.
It is. They observe a really comprehensive list of traits, covers the corm, the flowers, and the leaves. And these observations, they aren't just a single snapshot.
No.
No, they're made throughout the plant's entire growth cycle, and they're tied to specific developmental stages.
Okay, what are the key stages they focus on?
The guidelines highlight 3 main stages. Stage 30, that's flowering. Usually October, November, when the flowers are fully open, you can see the stigma and anthers.
Got it.
Stage 70 is the vegetative stage, typically around March. That's when the leaves are visible up to their full growth. And stage 90 is corm maturity, May, June time. The leaves have dried up and the new corms are mature underground.
So they're tracking changes right through the plant's life. How do they decide which specific plants to observe within those plots?
Good point. For the distinctiveness and stability assessments, they take detailed observations on 30 randomly selected plants, or sometimes parts of plants, like leaves or flowers.
30 plants per location.
Your location, yes. And those 30 are chosen by selecting 10 from each of those 3 replicated plots we mentioned.
Ah, random selection from each replicate. That's key for avoiding bias. Right?
Absolutely. Standard scientific practice.
And how do they standardize something like color? That seems like it could be subjective.
That's actually a great example of the precision involved here. For color characteristics, they use the latest Royal Horticultural Society color chart, the RHS chart.
Okay, what's that?
Think of it like a universal paint color swatch book, but specifically for plants, it's incredibly detailed.
So instead of just saying red.
Exactly. They'll use a specific code. Like the guidelines mention 41B235 for light red on the stigma, or 46A62 for dark red. Using these codes ensures consistency. It doesn't matter who's observing or where they are.
That completely removes the guesswork.
Amazing. Okay, now let's get right to the heart of it. The specific characteristics, the things they use to define a variety. Looking at the list in the source, the detail is just incredible.
It really is.
What are some examples that really stand out, or maybe reveal something surprising about saffron?
Well, the guidelines list, I think, 33 characteristics in total.
33.
Yeah. And they classify them in a couple of ways. There are qualitative characteristics, or QL. These have discontinuous states, like a specific shape or present absent, things not usually swayed much by the environment.
Okay, like it has this shape or it doesn't.
Pretty much. Examples given are corm tunic color or tepal shape. Then there are quantitative characteristics, QN. These show a range of variation, and they're recorded on a scale. Think length, weight, number of flowers.
Right, so QL is like, is it striped or not striped? And QN is more like, how tall is it on a scale? Exactly. QL traits are generally more stable against environmental influence. QN traits are still really important for DUS, but they're assessed on that scale, like short, medium, long. Now, rather than list all 33...
Please don't.
No, let's just pull out a few that really illustrate the depth of this process.
Okay, yeah, I'm really curious about what hidden identifiers they use.
Okay, here's one that might surprise you. The texture of the corm tunic.
The tunic. That's the papery coating on the underground corm, right?
That's the one. They classify its texture. It can be smooth and splitting, have interwoven fibers, or be wholly parallel. That's a qualitative trait. QL, observed at stage 90, maturity.
Wait, the texture of the coating on the underground part is a key identifier.
It is.
That's fascinatingly specific. I mean, I would never have thought to look at that. What does that detail tell them?
It's actually a surprisingly stable trait within a given variety. It's almost like an underground fingerprint. Another one related is the shape of the naked corm itself after you peel off that tunic. It could be elongated ovoid, flattened, or a flattened globose.
So you have to peel it.
To observe this trait, yes. These corm traits, shape and tunic texture, they're really fundamental to the variety's identity and maybe even its propagation potential.
So the part you don't even see when it's growing is vital for classification.
Yeah.
Incredible.
Hmm.
What about the flower, the part we actually associate most with saffron?
Oh, absolutely. Lots on the flower. For instance, they measure the length of the outer tepal, that's basically the petal,
Okay.
and they classify it quantitatively. Short, less than 3 centimeters, medium, 3 through 4 centimeters, or long, more than 4 centimeters. They also look at the tepal shape itself. Is it linear, obovate, oblanceolate, or elliptical? That's a QL trait.
And you mentioned earlier they even check the veining pattern on the tepals, like discontinuous, continuous, or parallel.
Yes, that's another one. Again, it sounds like such a small detail, doesn't it?
It really does.
But it's considered a stable trait that helps differentiate varieties. And of course, the stigma color is critical. That's where those RHS chart codes we talked about come in, defining that very specific shade of red.
Right, using those precise color codes for the actual saffron threads or the part that produces them.
Exactly. But perhaps one of the most revealing measurements is how they assess the pistil.
The pistil.
Yeah.
That's the whole female part, including the stigma.
Correct. It includes the style and the 3 stigmas. So after carefully removing the tepals and the anthers,
Okay.
they measure the total length of this pistil. That's a quantitative measure, short, medium, long, based on centimeter ranges. But they also measure its dry weight.
The dry weight of just the pistil. How on earth do they do that accurately?
They dry it in a vacuum dryer, very specific conditions, 60 degrees Celsius for exactly 25 minutes.
Exactly 25 minutes. Wow.
Then they weigh it and classify it as low, less than 7 milligrams, medium, 7 to 12 milligrams, or high, more than 12 milligrams. This is done back at stage 30 during flowering.
That precision, the exact time and temperature. Why? Why go to that length for the dry weight?
Well, think about it. This very exact method ensures they're comparing the true mass of the crucial saffron-producing part of the flower. It removes all the variability from just moisture content, which can change easily.
Okay, so it's a standardized comparison.
Right. And the dry weight of the pistil is thought to directly correlate with the concentration of those key compounds we value in saffron. Things like crocin for color, picrocrocin for taste, and saffronol for aroma.
So this test isn't just verifying identity. It's indirectly getting at the essence, the potential quality of the saffron that plant will produce.
You've got it. It connects that deep scientific detail directly back to the characteristics and the value we associate with saffron. It's not just about what it looks like. It's about measurable potential.
That is genuinely fascinating. It makes so much sense now.
And they look at reproductive traits too, naturally, like the total number and weight of daughter corms produced per plant by the end of the season, stage 90. That tells you about the variety's propagation potential, how well it multiplies.
So it really is a whole plant assessment across its entire life cycle. From the underground corm you start with, to the flower it produces, its quality potential, and even the next generation of corms.
That's absolutely right. It's incredibly comprehensive. And, to make the whole process of comparing a new variety against all the known ones more efficient.
Yeah, that must be a huge task.
It is. So they use certain key characteristics first to kind of group varieties that seem similar. It helps narrow down the comparisons needed later.
Smart. Which traits are good for that initial grouping?
The guidelines mention using characteristics like tepal shape and the shape of the tepal apex, the tepal's veining pattern, tepal color on the ventral surface, cataphyll color.
Cataphyll, what's that?
Those are the protective scale-like leaves around the emerging shoot, often whitish or brownish, observed at stage 70. And also those corm traits we discussed, corm tunic texture and the naked corm shape. These are generally traits considered relatively stable and maybe easier to observe quickly, helping to sort varieties into manageable groups first.
Right, it streamlines that massive comparison job.
Yeah.
And who are the people, the institutions, actually doing all this painstaking work?
Well, These specific guidelines, they were developed through a collaborative effort. There was a working group or task force committee and various contributors listed, experts from different Indian institutions.
So a team effort.
Definitely. And the official DUS tests themselves, they're conducted at specifically recognized centers. The guidelines name two: the Advanced Research Station for Saffron and Seed Spices, which is under Skust, Kashmir,
Okay.
and the CSR Institute of Himalayan Bioresources Technology in Palanpur.
So it's a national effort involving dedicated expert institutions.
Precisely. It really underscores the scientific infrastructure and the human expertise that's required to manage and protect these really valuable plant resources effectively.
So let's wrap this up. What does this all really mean? We've taken this incredibly detailed deep dive into these official Indian guidelines for testing saffron varieties.
We have.
And we've seen that defining a variety isn't just, a quick look. It's a truly rigorous scientific process, all rooted in proving those three key things: distinctiveness, uniformity, and stability, DUS.
Yeah, and you now know that proving a saffron variety's identity involves, well, everything from the specific size and health of the corm you plant
Right.
to multi-year testing, often at different locations,
Mhm.
and these incredibly precise measurements, things like the shape and texture of the underground corm,
Which is amazing.
the veining pattern on a petal, those specific RHS color codes, and even the dry weight of the stigma-bearing pistil, which hints at its potential quality.
Exactly. So hopefully you now have a much deeper appreciation for the sheer scientific effort and intricate detail that's required just to define and protect something as specific and valuable as one variety of saffron. It's built on dozens of these meticulously observed, measured characteristics tracked right across the plant's entire life cycle.
And maybe here's a final thought to leave you with. Considering this incredible level of detail, this complexity needed just to officially define and protect one plant variety like saffron, what does that imply about the hidden complexity and, frankly, the astounding diversity just waiting in the world all around us? Even in things we think are simple or familiar, perhaps there are layers upon layers of these intricate, measurable details that define their unique identity. Maybe there's always more depth waiting to be discovered, if we just decide to look closely enough.